So, hello, I am Marina Salvadores from France Supex Lab at the IRB of Barcelona, and I will be presenting our poster entitled as Redistribution of Mutation Rate across chromosomal domains in human cancer genomes. To give a bit of context, passenger somatic mutations accumulate during cancer evolution, and the distribution of these mutations are not uniform. There are parts of the genome that substantially accumulate more mutation than others, and this happens at multiple overlapping scales. To study the variability in this mutation rate at the megabase scale, we use a feature that we call regional mutation density, or RND for short, which is basically the count of mutations in 1 megabase windows along the whole genome. Previously, this regional mutation density has been correlated with chromatin accessibility and replication timing in a very tissue-specific manner. For this project, what we did is we calculate these regional mutation density patterns for a total of 4,000 tumors across 2,500 windows, and we apply a non-negative matrix factorization algorithm, which is a dimensionality reduction method, to extract regional mutation density signatures. These RND signatures captures the underlying mutation landscape that contributes to this final regional mutation density profile at different proportions in its different tumor. Once we apply this methodology, we obtain a total of 13 regional mutation density signatures, from which 10 of them were tissue-specific, like this regional mutation density scheme, while the other 3 are global, so they are universally present across tissues. The RND flat signature captures the known redistribution of mutations upon DNA mismatch repair failure, which are samples that are macrosatellite unstable. And this was known, so it serves as a validation that our method works. Apart from this, we found homolog repair deficient and samples with high apopec mutagenesis to show the same redistribution of mutations as previously observed in microsatellite unstable samples. This signature basically captures the increase of mutations in early replicating regions compared to the background, which ends up producing a flatter final regional mutation density profile. Apart from RND flat, we found 2 other RND signatures which are, to our knowledge, novel, and we name RND Global 1 and 2. In order to understand RND Global 1, what we did is we tried to explain this signature using the chromatin features that we download from ENCODE, and we calculate for each of these features their density across our 1 megabase windows. We try to predict the RND Global 1 from these features using the whole dataset, so it's sampled separately, fitted in the model, 1 sample at a time, or the average of the feature. And what we saw is that the RND Global 1 could be predicted from the variability within its feature, but not the average. In order to characterize this variability, we calculated replication time and chromatin remodeling signatures at this megabase scale. And we correlate them with the RND Global's signatures. What we observe is that the replication time, heterochromatin, and DHS remodeling signature that correlate with RND Global 1 are probably a mirror image of the same phenomena, that is the whole chromosomal domain switching on and off. Because as we can see, there are in these species the same peaks that we observe in our RND Global 1 signature. We also performed a causal analysis in which we found that RND Global 1 occurrence was associated with RB1 deletions. When we checked for the gene expression signatures at the level of the pathways, we saw that the relevant replication time DHAs and heterochromatin PC signatures show higher expression of cell cycle and E2F target genes, and the same gene expression signatures were found in RND Global 1 tumors and RB1 deleted tumors, which makes sense because RB1 is known to be upstream of the E2F targets. For RND Global 2, what we observe is that the mutation rates follow a distribution that resembles the canonical RND landscape, so increasing mutation density later replicating overall, except that we see a drop of this mutation in very late replicating windows. In specific, this RND Global 2 redistribution kind of linearize the association of RND to replication time by suppressing the more prominent peaks in regional mutation rates, but not affecting the minor peaks. For this signature, we tested for association of their activities with copy number and deleterious mutation and found that TP53 mutations to be strongly associated. As a support evidence, we see that the TP53 deletions, as well as amplification in non-oncogenes that phenocopy the TP53 loss, to be positively associated with RND Global 2 activities. So the take-home message will be that with our method we were able to identify 13 regional mutation density signatures that describe a global genome-wide mutation redistribution across many megabase size domains, and that we identified 2 novel global RND signatures that one of them show association with plasticity in DNA replication time and heterochromatin domains, and the other one a decrease of mutation rate in late replicating. And if you want to know more about this, you can please check our preprint by scanning this code.